Hi, my name is Nazatul Najwa Binti Nizam. My supervisor is TS Dr. Aisha Binti Muhammad Rehan. So this study is about antibacterial synergy effect of sea cucumber extract and Kelulut honey combination. The abstract, the antimicrobial susceptibility tests are used to determine the possibility of an antimicrobial agent to be effective in inhibiting the growth of bacteria in vitro under standardized experimental conditions. The antibacterial activity of Kelulut honey and sea cucumber individually and in combination were comparatively assessed with an established deficient ACEs and broth dilution method. Most of the samples exhibit small or absence of inhibitory activity using this division method. Only Kelulut honey sample with 100 concentration have positive antagonism effect against E. coli and S. epidermidis. For broth microdilution method, gram-positive bacteria was found to be more susceptible as compared to gram-negative bacteria species with lower minimum inhibitory concentration, are also known as MIC. So with lower MIC value of Kelulut honey, sea cucumber or combination sample. The growth of gram-positive S. epidermidis is inhibited by sea cucumber extract and at MIC value of 0.031 mg per ml, Kelulut honey at MIC value of 0.063 and combination sample at 0.063 also, all of which are of lower MIC value as compared to E. coli inhibition. The absorbance at optical density 600 nm wavelength was also measured to monitor the growth of bacteria in solution. The higher the OD 600 value, the higher degree of light scattered by bacteria in the solution, which indicate the higher bacteria count. Conversely, the lower the OD 600 value, the lower the bacterial count. From the result, broth microdilution proved to be a more effective method to assess the antibacterial activity in Kelulut honey and sea cucumber samples. Two combinations showed the best antibacterial synergy with OD 600 value of 0.000 against both gram-positive and gram-negative bacteria, which are first with half sea cucumber and half ratio of Kelulut honey combination, and second with 3/4 sea cucumber ratio of 1/4 Kelulut honey combination. Introduction. Honey is a natural substance with flavored viscous solution produced naturally by both honey bees and stingless bees. In vitro and in vivo research have revealed its antibacterial activities. Common honey producing bees in Malaysia include Heterotrigona, which produce Kelulut honey. Kelulut honey is more diluted compared to other types of honey. Kelulut honey has been claimed to have various pharmacological properties as antibacterial properties. Sea cucumber have leathery skin and gelatinous body shaped like a soft body cucumber. They are one of many marine animals with valuable nutrients and medicinal properties. The combination of sea cucumber extract with stingless bee honey holds a great potential for health supplement. However, the specific results of the combination have yet to be determined. For literature review, we will discuss about bacteria, natural antibacterial sources, synergy, combination of cucumber and Kelulut honey, and also antimicrobial resistance. For bacteria, disease may be caused by internal factors such as genetic deficiency and dysfunction of the autoimmune system, or by external factors, microorganisms. The use of antibacterial is very important in reducing the spread of infectious diseases. Second, natural antibacterial sources. Natural products are now being used as a preventative treatment as well as being integrated or complement conventional medical treatments. This is because natural products generally contain low toxicity and cause less side effects, with some having sufficiently high potency to prevent or overcome infections. In the treatment of different types of diseases, natural products have pharmacological effect that can be helpful. This will not only be active ingredients for traditional medicine, but also for modern medicines. These natural products are also used in chemical synthesis as the starting point for medicines discovery, from which synthetic analogy are developed with improvement effectiveness, strength, protection, and purity. For synergy, the method of identifying synergistic combination is dependent on trial and error. For testing synergistic effect, the isobel approach is the most convenient and experimentally acceptable method. If the effectiveness of the combination is more than the effect of either the agent alone or the sum of the effect of the individual agent, and antagonism is less than the expected effect of the combination, it is deemed synergistic. Next, combination of sea cucumber and Kelulut honey. The uses of combination honey and sea cucumber extract Heurig's cough syrup. The effect of the Heurig's cough syrup is claimed to be satisfactory as it would relieve cough and sore throat. The product from sea cucumber and honey started to increase in demand due to the discovery of its benefits. The combination of stingless honey and sea cucumber extract has yet to be studied. Fourth is antimicrobial resistant. Next is antimicrobial resistant. The antimicrobial resistance is a global health crisis in this present time. This misuse and unprofessional use of antibiotics have caused a post-antibiotic time where the existence of antibiotic protection could become negligible. The antimicrobial resistance can affect people regardless of the age. Common antibiotics such as are losing their efficiency due to the increase of microbial resistance. And antimicrobial resistance can be caused by many factors, includes mutations and resistant gene transfer in bacteria, poor regulations over prescriptions and inaccurate usage of antibiotics, lack of awareness on the usage of antimicrobial, and overuse of antibiotic in poultry and livestock instead of focusing in infection control. Last, antimicrobial susceptibility test. The antimicrobial susceptibility tests are used to determine the possibility of an antimicrobial agent to be effective in inhibiting the growth of bacteria in vitro under standardized experimental conditions. The antimicrobial activity can be described in terms of zone of inhibition in a millimeter of bacterial growth surrounding a disk that contains antibiotic agent, or as a minimum inhibitory concentration MIC in a unit of mg per ml of the antibiotic against the bacterial activity. The determination of antimicrobial activity can be performed with different methods such as broth or agar dilution, disk division assays, and gradient division method. Methodology. Both sea cucumber and Kelulut honey are obtained from extracted ready-made commercial product and have been extracted in a liquid form. For sea cucumber, it is obtained from Selera Suarasa Food Sungai Petani Kedah, while for Kelulut honey is obtained from Madu Kelulut Asli Rimba Kahang Johor. So in this study, we use 3 different methods to test the antimicrobial activity, which is first is disk division method, and second is antimicrobial which is second is broth micro dilution method. So the media that we use in this study is nutrient media, nutrient agar, and also nutrient broth, while for bacteria, E. coli was used as a gram negative bacteria, while S. abnormalities was used as a gram positive bacteria. So for this division assays, the method was summarized as in the flowchart, where first the samples was dilute with sterile distilled water with various concentration, and then next 80 microliter of diluted samples was prepared with a 6 mm filter paper disc, and then third 100 microliter of vinculum was sprayed on neutron agar plate, and then it is transferred filter paper disc to the media. So for this method, ampicillin antibiotic was used as a positive control, while sterile distilled water was used as a negative control. After all the procedure done, the media was incubated at 35 to 33.7 degrees Celsius for 16 to 24 hours. So for the next method is broth microdilation method, where the method was summarized as shown in the figure. But the dilution that we made is only until 1/68 with the test tube of egg. So all the test tube 1 until test tube 8 was filled with 1 ml of samples, and then for test tube 1, except for test tube 1 where it is filled with 2 ml of samples. So for the positive control, which is test tube 11, it is filled with 1 ml broth and also 1 ml bacteria, while for the negative control, it is filled with 1 ml honey and also 1 ml broth. So and then the bacteria, 1 ml bacteria was filled into the second test tube, and then from the second test tube, we take 1 ml from the second test tube to transfer to the third test tube, and then dilute until the 8th test tube. Result and discussion. The result was summarized as shown in the table. So for first method, which is this division method, most of the sample did not exhibit inhibition zone, or just a small inhibition zone, except for the Kelulut honey with 100% concentration, which E. coli, diameter inhibition zone for E. coli was 12 mm, while S. apernabis was 70 mm. This is not in line with the theory because of some factor. One of the factor is the sample, which is cucumber sample, has been stored in the refrigerator since March 2021. Thus, the length of time taken may have reduced the antibacterial effect of the sea cucumber extract. However, the supplier of this iGamat claimed that the testimonies from their buyer over the years have indicated that the coalomic fluid extracted has beneficial properties. Two recent publications have reported on the antimicrobial activity of stingless bee honey from T. carbonaria, one reporting no antimicrobial activity, whilst the other showed considerable activity. In the current study, little activity was seen by agar division, which concur with one of the previous study. This include insensitivity. The factor can be included as a insensitivity, whereby low level of antimicrobial activity are not necessarily detectable, and variability, whereby minor variation in experimental conditions such as agar volume, inoculum concentration, and incubation condition may cumulatively lead to significant variability in result. From this study, gram-positive bacteria were found to be more susceptible than gram-negative bacteria in previous study. Also shown the same. So for broth microdilation method, it is more effective compared to the distribution axis, as the result shown in the table. And then, but however, the optical density measurement also have some limitation. Turbidometry detects only the upper part of microbial growth curve and require calibration in order to correlate the result with viable counts obtained on media. Most of the sample exhibit small or not any inhibitory activity for method of these deficient assays. Only Kelulut honey sample with 100% concentration have positive antagonism effect against E. coli and S. aprenebilis. For both microdilution method, gram-positive bacteria is shown to be more susceptible compared to gram-negative bacteria species. The higher the OD600 value, the higher degree of light scattered by bacteria in the solution, which indicate the higher bacterial count. Conversely, the lower the OD600 value, the lower the bacterial count. For the best ratio concentration of antibacterial synergy, two combinations showed OD600 value of 0.000 against both gram-positive and gram-negative bacteria, which are concentration of two different concentrations, which are first with 1/2 sea cucumber of ratio 1/2 Kelulut honey combination, and second 3/4 sea cucumber ratio of 1/4 Kelulut honey combination. From the result, both microdilution proved to be a more effective method to assess the antibacterial activity compared with these deficient assays. Antibacterial assays against multiple bacterial species should be performed to determine the range of its antibacterial activity as well as against known resistant bacterial strains. Thank you.